All right. Thank you everyone for joining us today. I'm thrilled to help introduce today's webinar focused on the strategic West Central water type and eDNA assessment presented by Jamie Glasgow of Wild Fish Conservancy. My name is Rebecca Brown and I work for the Department of Natural Resources, where I am part of the National Estuary Program Habitat Strategic Initiative Lead Team, or Habitat-SIL, supporting the land development and cover implementation strategy. So first, we've got a few quick housekeeping notes. Today's webinar is being recorded and it will be available in the not so distant future on our website. So I guess stay tuned for that. And we do hope to have time for questions at the end of the presentation. So when we get there, we'll ask you to type a question in the chat box and I'll facilitate a question with Jamie. And then if you're having Zoom issues, please contact Devin Rostifer. She's on the call here, so you can reach her in Zoom chat, or you can also email her, and her email is on the screen. So as I mentioned, I work for the Habitat Strategic Initiative Lead Team, which is an interagency team supporting the National Estuary Program, co-led by Washington Department of Fish and Wildlife and Washington Department of Natural Resources. Our focus is on advancing Puget Sound recovery through collaborative planning and strategic investments. We award grants of the Puget Sound Geographic Funds through a cooperative agreement with EPA in support of the Puget Sound Action Agenda and implementation strategies. One of the reasons this is exciting is because these dollars can go towards traditionally difficult to fund actions that catalyze greater opportunities for recovery. So today you'll hear from Jamie Glasgow, the Director of Science and Research at the Wild Fish Conservancy. Jamie has been conducting fisheries research and restoration projects in the Northwest since 1996 and is instrumental in data collection, analysis, and reporting aspects of most Wild Fish Conservancy projects. Today's presentation will describe how Wild Fish Conservancy expanded water type and environmental DNA assessments in West Central Puget Sound watersheds to address data gaps crucial to the effective implementation of critical area ordinances, habitat restoration planning, and salmon and steelhead recovery planning. The project provided ground truth data on the location and classification of streams within prioritized watersheds, along with fish species-specific presence data to enable protection and restoration efforts to be directed towards areas where actions will have the greatest impact. The results of this project contribute to the land development and cover implementation strategy by providing local governments with better data, including improved regulatory maps, which will help them better protect critical areas. This project was identified by our Habitat Strategic Initiative Advisory Team as an important contribution to the land development and cover implementation strategy and the 2018 to 2022 Puget Sound Action Agenda. It represents a longstanding continuation of the Habitat-SIL's work to catalyze recovery actions for habitat and critical species by using the best available science, local knowledge, and partnerships to inform recovery plans. Even though the funded portion of Jamie's project has come to a close, we are very interested in the discussion today and continuing this conversation for how efforts like this can inform regional recovery goals. So one more note that if you want to get more information on the Habitat-SIL, please visit our webpage and you can subscribe to get updates. And then I'll drop some links in the chat when I'm done with this little spiel. And then I look forward to this discussion today, and I've shared my information here, so if folks want to reach out, feel free to. And then with that, it's my pleasure to introduce Jamie Glasgow. And Jamie, I'm going to stop sharing my screen now, so you can go ahead and share yours.
Great, thank you, Rebecca. Okay, good morning, everyone. Thanks for taking time out of your day to hear about this important project. Again, my name is Jamie Glasgow. I'm Director of Science for Wild Fish Conservancy. We are a science-based conservation organization, a nonprofit, and we do work throughout the Northwest to preserve, protect, and restore wild fish ecosystems, largely through science, education, and advocacy. And today, I'll be presenting the final project presentation for our West Central water type and eDNA, or environmental DNA assessment, which was a near-term action which the partnership funded. This was a 4-year-long project with a lot of different components, but I've simplified it for this presentation into kind of 3 main chapters or categories, and that's what I'll use to organize today's presentation. We'll start with the component which involved surveying and water typing 5 prioritized West Puget Sound watersheds, and then move on to filling critical data gaps about the distribution of fish species of interest throughout West Sound. That's using environmental DNA. And then finally, how we worked to update federal, state, and tribal databases with the accurate and ground truth data that we collected over the course of this project. So there's a lot of people on this webinar, and some of you are going to be more familiar with water typing than others. So I'm going to take just a step back and start at the very beginning for water typing and make sure we're all on the same page about what it is and why it's important. So bear with me, those of you who know all this already. But water typing is simply a classification system that Washington uses to inform land use around streams. It really is answering the question, where are the streams and where are the fish habitats within those streams? This is a process which was originally developed by the State Department of Natural Resources in the mid-70s, specifically for forest practices and regulations. It's spelled out in Washington Administrative Code, and you can reference that at any point in time. And it identifies 4 main categories or types of stream reaches. Type S streams are shorelines, which are actually managed by Department of Ecology, not by Department of Natural Resources. And type F streams are fish habitat streams. Type N streams are non-fish habitat streams. And type U streams are streams that exist, but we don't know whether they provide fish habitat or not. And each of these different types then receives a different buffer size depending on the land use or jurisdiction. But generally, they go from larger buffers to smaller buffers as we work our way down this list of SF, N, and U. So as I mentioned, this system was originally developed by DNR for forest practices, but it was subsequently adopted by most state and local governments to identify and classify streams for planning purposes. Just as a quick example, because much of our work was in Kitsap County, I'm providing the Kitsap County Critical Areas Code development standards, which here you can see link the water type, F, N, P, or NS, to different buffer widths, which are expected or required in Kitsap County. Just a real quick note that sub P and sub S for type N streams stands for perennial or seasonal. The state defines fish habitat or type F waters in one long and awkward sentence, which I'm going to read here because it's really important, and I think a lot of people may not be aware how the state defines fish habitat for the purposes of water typing. And again, this is in WAC 222-16010, and it says type F waters provide fish habitat defined as habitat which is used by fish at any life stage at any time of the year, including potential habitat likely to be used by fish, which could be recovered by restoration or management, and includes off-channel habitat. Did I mention it was awkward? So this covers a lot of ground. It's a pretty conservative definition of fish habitat, which we're glad to see. And it's important to note that it includes streams that may not be currently occupied by fish. It also includes seasonal streams that could be dry during parts of the year, but used by fish at different times of the year. So Department of Natural Resources provides maps online that differentiate the classification of stream reaches, like this one. So here you can see in red, DNR is showing their understanding based on a model of where the type F stream reach is in this small watershed. and yellow reaches are type N non-fish habitat reaches. So local and state agencies, in addition to DNR, would use this information to require buffers on streams. As I mentioned, type F streams get bigger buffers than type N streams. So we've all seen old photographs where Washington's streams didn't always get the protection they should have. We know that riparian areas are important and that we can't afford to lose them, especially on fish-bearing stream reaches. And this is exactly what the water typing system is designed to prevent. However, there are unfortunately many recent examples where that doesn't always work. The system isn't always working. And that's because regulatory maps that guide stream protection, water type maps, are inaccurate. The maps frequently underestimate the distribution of streams and fish habitats, and many streams are incorrectly mapped or are not on the maps at all. This is because the stream maps are modeled and not always ground truthed. And as we all know, models are not infallible. As a result, misidentified fish habitats are unlikely to receive the protection they warrant under existing laws. So we've got good rules in place to protect streams. They're driven by water type, but the simple issue of the maps being inaccurate can lead to situations where streams aren't receiving the protection they should be. To demonstrate that point, let's go back to this same watershed. This is Cowling Creek in Kitsap County. And here again is DNR's understanding of this watershed. These are GPS points our survey team collected during the course of the water type assessment of Cowling Creek. And here's the actual extent of the Cowling Creek watershed. Again, red indicating fish habitat, yellow and indicating non-fish habitat. So in this case, miles of stream that were unmapped, they may or may not be protected during land use applications in this upper watershed, but certainly for planning purposes, it becomes difficult, if not impossible, without having a good sense for the actual extent of this watershed. That means that streamside buffers may not be receiving the protection they warrant. I want to mention too that the implications of these inaccurate maps goes well beyond riparian buffers. For example, a barrier culvert at this location might be overlooked due to the perceived small amount of fish habitat believed to be upstream from it. So when riparian corridors are not adequately protected, there are significant consequences. There's impacts to the hydrograph and erosion and water temperature and reduced water quality, all of these resulting in simplified in-stream habitat and implications for fish populations that we're trying to recover. For more information on the importance of riparian habitat, WDFW provides 300 plus pages on the virtues of streamside trees. In this document. There is a volume 2. I'd ask that you read both of these online and don't print them. The bottom line is riparian corridors are an integral part of watershed health and will become more so due to impacts from climate change. That is one reason that Wild Fish Conservancy has for over a decade now been performing systematic water type assessments throughout Western Washington. This map shows the extent of the surveys we've been performing. So the project I'm presenting today is just one part of many other projects where we've been walking streams and mapping and classifying them using the state's methodologies. For this project, we focused on 5 West Puget Sound watersheds. These were identified with input from the West Sound Partners for Ecosystem Restoration. They're where we had data gaps and where we expected increased land use pressure through growth and forest practices. To begin with, we built a GIS and used LiDAR and flow accumulation modeling to identify potential streamside landowners in each watershed. And we sent each of them a self-addressed stamped letter explaining the project and requesting their permission for our survey crews to access the stream on their property. We built GIS to track landowner responses and incorporated the permission GIS onto the data collection tablets our crews were using to collect data in the field. So over the course of the project, we sent permission request forms to 2,465 parcels within these 5 priority watersheds. Of all of those, 706 landowners replied, and of the 706, 424 granted access to a Wild Fish Conservancy field staff, allowing us to survey or access streams on a total of 2,300 acres of private land. We certainly would have liked to have seen a greater response, but expect that the COVID pandemic played a big role in the limited response that we got to our request. Within these 5 watersheds, then, our staff over the course of this project collected 1,280 GPS points and over 4,000 photographs. At each of the GPS points we collected, we followed the state water typing protocols to collect bankful width, wetted width, and gradient data on notes about the quality and character of the in-stream and riparian habitat, and descriptions of habitat protection and restoration opportunities that we documented over the course of the project. We also dip netted and electrofished to determine fish presence and identify species and then release them. These species included chum and coho and Oncorhynchus micus, which could have either been juvenile steelhead or resident rainbow trout, cutthroat trout, juvenile trout that were too small to differentiate as either cutthroat or steelhead or rainbow, brook lamprey, and sculpin species. So during the course of the project, we captured, photographed, and released 202 fish. All of this information that we collected is geo-referenced in an interactive web map that's available on our website. The GIS driving the map are available for anyone to download. And I am hoping to have some time at the end of the presentation to walk you through how to access and use this interactive web map if you're not already familiar with it. So I'm going to shift gears now to the second chapter, which is the environmental DNA, or eDNA, which we use to document the presence of species of interest in streams where they had not been previously documented. eDNA is an extremely effective tool for determining the distribution of species of interest. It's innovative, but it's not new. I suspect most, if not all of you, have heard of it and maybe even have used it. There are now hundreds of published papers and even an entire journal dedicated to eDNA research. One of the reasons it's gained so much popularity is that it is very effective, and more so in many cases than traditional sampling, especially at detecting the presence of rare or cryptic fish species. Here's just a couple of relevant papers where eDNA was used to identify just those rare and cryptic species. Unlike conventional methods, eDNA is harmless to the fish and therefore has, in many cases, advantages over conventional sampling, even beyond the increased detectability of species of interest. So for years, Wild Fish Conservancy has been working with the U.S. Forest Service's National Genomics Center. And in this project, we used their methods and equipment and their laboratory for our eDNA analysis. The process of collecting eDNA data involves pumping, in our case, 5 liters of water through a filter. The filter strains out the particles that are in the water, including DNA. The water goes right back into the stream at the sample site, and the filter comes back to the lab with us. We follow a very strict contamination procedure to avoid contaminating the samples with DNA that may have come from other sources besides the 5 liters of water that we're collecting at the site. and the laboratory receives our samples and archives half the sample and cuts the other half of each sample into thirds for triplicate analyses. So over the course of the project, we identified 173 sample sites at locations in West Puget Sound where species of interest hadn't previously been documented. You can see we sampled for Chinook presence, coho, Oncorhynchus mycus, again, either rainbow trout or steelhead, and then a marker for any salmonids and any sculpin. A conservative rule of thumb for eDNA is that you'll get a positive result if the species of interest was present at or upstream from the site you sampled within 1 week and within 1 kilometer. So as with our water typing results, we've built an interactive web map for our eDNA results, which is also available on our website, and which I also hope to be able to show you at the end of this presentation. And with this tool, you can use the layers menu, which you see in the upper right-hand corner, to select your species of interest and see where we sampled for that species and within those sites where we detected its DNA. So for example, with coho selected, you'll see we you'll see where we found coho DNA as the green pins on this map, where we didn't find it as the white pins. And then the white circles are simply where we collected eDNA samples, but we did not test for coho, most likely because they had already been documented in those streams. Zooming in a little more and clicking on a point, you'll see we have data available for each eDNA point, including the sample date, the filter volume, and then notes and species tested for and the test results. On to the third chapter. This is possibly the least interesting, but maybe the most important part of the project. Because our field verified data and mapping improvements are only useful if they are used. And so we made sure to submit our results using the processes developed by the agencies that manage this sort of data. And that's what I'll describe briefly. Next. I've got 4 main different audiences for the data sharing. The first is the wide world, which we've attempted to capture with our interactive web maps. The second is the Department of Natural Resources, which again manages water typing. The third is the Washington Department of Fish and Wildlife and the Northwest Indian Fish Commission, which together manage the statewide integrated fish distribution database, or SWIFT. And the fourth is the USGS National Hydrography Dataset, or NHD. I've already mentioned these interactive web maps, so I won't spend more time on them here. These are the eDNA and water typing interactive maps that we just discussed. On to the Department of Natural Resources and their water type data management. DNR's water type modification process requires water type mod forms with detailed information about fish and fish habitat observations. So all the data we collect in the field and the photographs get packaged into a required format that the Department of Natural Resources then reviews along with some or all of WDFW, the Department of Ecology, and the tribes. And so between March of 2021 and September of 2022, Wild Fish Conservancy assembled and submitted 100 water type modification forms in RIAs 14 and 15 using DNR's forest land and non-forest land water type mod form process, depending on the location of the proposed changes. Most of these mods or modifications were for streams where Wild Fish Conservancy had documented fish presence in reaches that had been incorrectly designated as type N or in reaches that were previously unmapped. So summarizing the results of that effort, these map and classification corrections are now reflected on the state's regulatory water type map managed by the Department of Natural Resources, but used statewide by state, local, and federal government agencies. So we submitted 16.5 miles of new type F habitat. 11.5 of those have already been incorporated. There are 5 more miles that are pending. We have upgraded almost 4 miles of type N water to type F water. 3.5 of those miles have been incorporated already. We have corrected the channel location of 5 miles of streams and added 0.6 miles of new type N habitat, and then removed 2 miles of streams that had been on the Department of Natural Resources regulatory water type map, but were not found to exist on the ground. Shifting over to the Department of Fish and Wildlife and the Northwest Indian Fish Commission, the statewide integrated fish distribution data is another data set which we updated with our information. This fish distribution data set is managed by DFW and the Northwest Indian Fish Commission to create a common data framework for fish information that better serves the management, conservation, and restoration of the state's fish habitat and fishery resources. Species-specific SWIFT data are available to the public through the Department of Fish and Wildlife's Salmonscape mapping portal. And these are also data you can download from the Department of Fish and Wildlife and incorporate into your own GIS. In this map, you can see the location of coho and coastal cutthroat trout that we captured and released that were found to exist outside of where they had previously been recorded in SWIFT. And so we shared those coordinates and the data from those surveys with the Department of Fish and Wildlife in order to add a total of over 18 miles of coho streams and almost 62 miles of cutthroat streams. This was based on 126 coho and cutthroat fish in hand observations that we made in stream reaches where they had not been documented in SWIFT. Finally, the NHD dataset, which is managed by the USGS, provides the most up-to-date and comprehensive hydrography for the nation. The Department of Ecology manages NHD in Washington. We used their NHD markup tool to submit 24 stream reaches that had been missing in NHD, but in which we documented fish. This will equate to 9.8 miles of new NHD stream channels once the NHD review process is completed. So I'm going to start wrapping up here and was asked to share some observations and opportunities that that came out of this work that we've done. So we'll just work our way through this list. The first, and it's one I started with at the beginning of the presentation, is that modeled regulatory water type maps provided by the Department of Natural Resources often underrepresent the upper extent of fish habitat, and many streams are mismapped or are not mapped at all. Accurate water typing and other stream mapping data are fundamental to the effectiveness of our forest practices regulations, our critical area ordinances, and other land use and habitat restoration planning and permitting efforts. Number two, submission of water type mod forms after water typing surveys is not required. The same is true for SWIFT and NHD updates. We feel like this is a missed opportunity. Entities that collect relevant data that could improve the state's maps are not necessarily sharing them. Widespread submission of ground truth, water type, SWIFT, and NHD corrections from foresters and consultants and NGOs and tribes and state agencies would improve the maps and reduce duplication of effort, and finally provide some quality assurance and accountability for the decisions that are being made using those observations. The third is a recommendation that Washington Department of Natural Resources should transition from their outdated and unique hydrography to NHD, or something that's more widely used and accurate. As we understand it, this transition is actually underway, so stay tuned. I believe that within the next year or two, we should see improved NHD hydrography. However, note that the actual location of streams and the distribution of fish and fish habitat will only be achievable through ground surveys. Number 4, the uppermost fish found on one day of the year may not represent the upper extent of habitat used by fish at any time of the year. You'll remember that the state's definition of type F, or fish habitat, is that habitat used by any species of fish, any life stage, any time of the year, including recoverable habitat. And so given the state's definition of fish habitat, and given the proliferation of barrier culverts and the depressed status of our fish populations, we believe water type surveys should rely more on physical channel characteristics and less on the uppermost fish found on the one day that a survey is being performed. DNR is in the process of updating its water typing rule to address this and some other problems. And then finally, an observation that WDFW could be a more appropriate manager of water type data in Washington. We believe this is true given the purpose and intent of water typing and its widespread use and implications outside of forest practices. The Department of Fish and Wildlife is the state agency charged with managing the health of Washington's fish populations and the habitats they rely on. WDFW also manages the state scientific collection permits required for water typing. Water typing is fundamental to the department's mandates and its expertise. So it looks like, I'm looking to you for a second, Rebecca, it looks like we have a little bit of time. I may shift gears and go over to our interactive web maps and just give a quick run through and a bit of a tutorial on how they work and how folks can access and use that information.
Yeah, that sounds great, Jamie. And then we should probably still have time for the questions that are starting to pop in the chat.
All right. Bear with me here. Okay, so I am on our Wildfish Conservancy website, which is simply wildfishconservancy.org. And in order to navigate to these interactive web maps that I've been talking about, if you go to resources, you can drop down a menu and find interactive maps there. And here you'll find links for the 2 interactive web maps we've been discussing. I'm going to start with the water typing map, which brings up this page. This is a revamped interactive web map. So for those of you who have been using our maps in the past, this will look a little different. It works in much the same way. We think it's a little more clear and user-friendly. I hope you do too. We start with a splash screen, which provides a link here for more information about water typing, including a short video about what it is and why it's important and the projects that Wildfish Conservancy is doing. Of course, some information about how this interactive web map tools, how to navigate through it, how to zoom and pan. And then an explanation for the different links which are available on the web map in the upper right-hand corner, for example. You can turn on and off the legend, the background maps. You can use a list of bookmarks for various projects, and you can search addresses, and you can measure distances. Here also is a link to download the GIS data that run the map, so you can pull this information and build your own GIS with it. And then once you click OK, if you need to see those instructions again, you can access them by clicking this information button down here in the lower left-hand corner. I'm going to use my mouse wheel to zoom and left click and drag to pan. And you'll notice as we change our scale, more information becomes available. So you can navigate to a stream of interest and recall that each one of these points represents a GPS point where we've collected information. Here we are in Purdy Creek, for example. If we scroll in even further, we'll see as dashed lines the DNR hydrography for this watershed. The solid lines are the Wild Fish Conservancy hydrography. And just as an example, here's a right bank tributary to Purdy Creek, which was previously unmapped. And then to see examples of the information that we collected in order to add this stream to the maps, you can click on the point, which opens up some more information in this left pane. At the top, which has its own scroll, you'll see some measurements of the habitat, the channel measurements, in-stream structures that were observed at this location, a description of it, a field call status on the passability, the fish passability of this structure, and then additional notes. You can get additional stream details that go beyond those point details by clicking this link, and then just click off that window to close that. And then here are the photographs that we collected at this particular location. And if you click on any one of the photographs, you'll be taken to a larger scale view of each photograph with the caption underneath, and you can scroll through each of the photos to see what our field staff saw at the time of the survey. I'm going to close the legend just to give ourselves a little more room. I want to point out one additional change from how our maps worked in the past. There are two different phases of water typing data represented in these maps. The information we're looking at here and the information we collected through this project on the 5 prioritized watersheds represent phase 2 point details. And here I'm in the upper left-hand corner, showing that we've selected phase 2 point details to look at these blue and red points. If we go to a different part of the state where we did surveys much earlier, you'll see the points are purple, and those represent phase 1 point details. And so in order to be able to see the data from those points, when you click on a point, you won't see the data until you click phase 1 point details. And then you'll be able to see the information we collected during that phase of the project. So if you're using this map to look at data above and beyond or outside of those collected during the course of this project, you'll need to be cognizant of which phase of data collection you're looking at, and that you determine by the color of the points you're clicking on. Okay, I'm gonna move on from the water typing map and take a look at the eDNA map, which is the link on the left here from our website, same jump off. And that will take you to a map that looks like this. I'm actually going to close this and do this again so you can see the splash screen. So here again, you can hide this splash screen, so you only have to read it once. But what it's reminding you is that you really don't get much use out of this site until you select a species of interest. And I want to point out that we've got a lot of different projects represented on this map. And so you'll see species that we did not sample for during this particular NTA. I also want to point out that this list is not complete here. You need to scroll down to see the complete list, which includes some freshwater mussels and Olympic mud minnow surveys that we've done. In my experience, it's much recommended to select just one species at a time. And when you do, you'll see here, I'm going to select Chinook. And here over the kind of landscape scale, you can see where we have sampled for Chinook eDNA, shown as a teardrop pin, where the sample was positive for Chinook DNA. Those are the green points. And then where it was negative, those are the white points. And again, these closed circles here are eDNA sample sites where we did not test for Chinook. Pretty self-explanatory once you've used it once. You have different layers that you can turn on and off. Some of those only become activated when you're zoomed in far enough. For example, we can turn on the water typing data that Wild Fish Conservancy created if you're interested in seeing a more accurate representation of the watersheds where we sampled for eDNA. You can also turn on the SWIFTI, or the Statewide Integrated Fish Distribution Data Set, to see where different species have, specific species have been already documented or are presumed to be present, either for spawning or rearing. And that legend is updated on the left here. Okay, I'm going to shift gears one last time and finish with a few slides, and then we can open it up to questions. Actually, just this one slide. Here's my contact information. I'd love to hear from you if you have questions about what we've done. Also, want to mention that we covered a lot of information in this presentation, but all of it and more is included in more detail in a final report, which we've written. That report will be on the Puget Sound Partnership's website and Wild Fish Conservancy's website shortly. You can always contact me if you can't wait. I'd like to acknowledge the Puget Sound Partnership for funding this near-term action, and also the landowners who gave us permission to collect data from the streams running through their properties. And I'll end by saying for any partners who are interested, we are actively seeking opportunities to expand this important work. These ground truth maps and fish distribution data are really fundamental, I think, to effective recovery planning. And we would like to see more, if not ultimately all, of Puget Sound's watersheds mapped in this way. With that, I'll stop and see if we have some time for questions.
Yeah, I'd say we've got about a little under 15 minutes for questions. So first, Jamie, thank you. That was great. Super interesting. And I also want to point folks to the chat, because there is definitely a conversation that's happening there too. But I think that I am going to just focus on some of the questions that have popped up. Okay, so how does the volume of water in a stream influence eDNA?
So the volume of water sampled is just one of several variables that affects your kind of detection probability with eDNA. There are trade-offs. The more water that you filter, the more noise there can be in the results. So you're looking for a sweet spot that allows you to collect the amount of water that leads to accurate results without overburdening your filter. If, for example, if a stream is turbid, it can take 3 or 4 filters to pump even just 5 liters. If we had to collect 10 liters, then we'd be there for an hour or two, and there are trade-offs again with the accuracy of the results when you have too much material on a single filter. If you were after a species that you think might be present but in extremely low abundances, you would be recommended, I have no doubt, by the National Genomic Center to collect more than one sample at 5 liters. So maybe 2 or 3 5-liter samples. But 5 liters is the sweet spot that particular lab, and this is at the National Genomic Center, has found based on their experience working with eDNA and fish species.
Great, thanks. We've got another. So really like the idea of eDNA and the non-invasive nature of it. How do you distinguish between reaches actually used by fish and those where birds or other species are eating baby fish and dropping remnants and defecating fish DNA?
Great question, and a real common one. It's important whenever using eDNA to not lose sight of what it is, and that particular question kind of hits the nail on the head. All eDNA is telling us is whether the DNA of a species that we've tested for is present. Yeah, it doesn't always mean that that fish, for example, is at or upstream from the site. It could be the result of a carcass that was dropped by an eagle or any number of other explanations. And so the advice that we give and that we've gotten from the National Genomics Center is that when you get a result that you weren't expecting, sample again, let some time pass, sample again. That's number one. Number two is, and we often do this as well, think of eDNA as a screening tool. And if you are seeing results that you weren't expecting, you might switch over to conventional tools at that location. Maybe you do a snorkel survey or some electrofishing or some trapping and see if you can bring the species of interest to hand. That in conjunction with multiple sampling can help weed out those anomalies and those one-offs that don't really represent what we're looking for.
Great, thank you. And kind of a follow-up on that, did your survey teams conduct electrofishing surveys and collect eDNA data on the same stream, same day? And are there logistical issues such as site contamination, site disturbance associated with simultaneously conducting e-fishing and collecting eDNA?
Absolutely. A lot of our field project time is spent coordinating so that we have at least a week and a half and ideally 2 or more weeks in between when we collected the eDNA sample and when we know anyone was in the stream upstream from where we're collecting that sample. So it is important to try to avoid those kinds of contamination issues. But it's also important to remember that it would be fine if somebody was in the stream upstream from where you took your sample, so long as they didn't have the DNA of the species of interest on their waders or wading shoes, for example, in a volume sufficient enough and likely enough to be captured by the eDNA sample you're collecting somewhere downstream. So there's a lot of research kind of looking into that risk of contamination and how much of an effort needs to be taken to avoid it. But from our perspective, we did everything we could to make sure that we were sampling at times and locations where there were not people in the stream upstream from us.
Great. So we've got one here. It says, great project and excellent presentation. Thanks, Jamie. How does 20 to 30% access permission to streams influence your stream typing findings? And how do you work around this constraint?
Yeah, so more is always better when it comes to access for water typing in particular. That was on the low side. And like I said, I think a lot of that had to do with the fact that we really started this field work in about the same time that the COVID pandemic was really ramping up. The good news is that with water typing, we can still accomplish a whole lot even with little access. The results are much, there's more depth to the results when we have more access because we're seeing more of the stream. But even with limited access, for example, at county road crossings, we can collect information about fish presence or the presence of fish habitat, and that information then can inform the water type from that point downstream to the next known location of fish habitat. So just as a quick example, even if we had no access from landowners in a watershed, if there was a road crossing near the headwaters of that stream, and we found fish there, we could type that entire stream from that location down to Puget Sound as type F waters, because it's presumed that wherever you find fish, they have access to habitats downstream from that location. So more is better. We were still able to accomplish quite a bit. You can tell by the distribution of the GPS points that we collected that we covered a lot of ground, and what we lose with less access is the ability to document stream features and characteristics, especially from our perspective, those that are important for habitat restoration and protection opportunities. And that's a real kind of benefit that comes out of these projects that's different than a lot of other types of projects. Quick example, when folks do a fish passage inventory, more often than not, it's based off of a GIS that looks at road stream interfaces. That's where culverts are. But I'm telling you a couple things. One is that stream layer that you're using is probably incomplete. So there's other stream road interfaces that aren't on your GIS. Number two is when you focus on the road crossings, you're missing features, natural or anthropogenic, that exist between road crossings, things like old derelict water withdrawals. Those are things that our team is able to observe and document and photograph. And those have huge implications for prioritizing fish passage restoration projects at culverts, because we don't want to do things out of sequence and spend a lot of money fixing fish passage restoration upstream from what turns out to be a barrier derelict water withdrawal dam that nobody knew existed. So there's big advantages to walking as much of these streams as possible. It's just hard to do.
Yeah, but I bet it's lovely out there to do it. What percentage of the survey area is urban and which percentage is commercial forest land?
Yeah, so our project really focused outside of, certainly outside of large commercial forest land. There were landowners within each of the watersheds that qualified as well, in DNR parlance, for forest practices, water type mod forms, as opposed to non-forest water type mod forms. And to put a percentage to it, I would say that, let's say that about 70% of the parcels that we collected data on were not forest land, and the other 30 were.
Great, thank you. Have you found fish in streams smaller than DNR's minimum bankful width criteria for water typing? And how about maximum gradients where you found any fish?
Yeah, great question. So little more background there. DNR in the WAC that I mentioned, 222-16031, DNR identifies, the state identifies default physical criteria for fish habitat. And this includes a bankful width of 2 feet and a gradient equal to or less than 16% or 20% in larger watersheds. That's larger than 50 acres. So just put more simply, if a stream channel is 2 foot bankful and less than 16% gradient, it's presumed to be fish habitat. However, the State Department of Natural Resources does allow surveyors to go into such a stream, electrofish on one day, not find fish, and identify that stream reach then as type N non-fish habitat. So there are default physical criteria that can be overridden with protocol electrofishing surveys, which have to be performed using a protocol provided by the State Department of Natural Resources in Forest Practices Board Manual, Section 13. So going back to the questions, there are definitely instances when we're finding fish, juvenile coho, for example, in stream channels that are less than 2 foot bankful. So those channels would not qualify as default physical criteria for fish habitat. However, fish presence always supersedes the default physical criteria. So anywhere fish are found is fish habitat, is type F. If you're not electrofishing, you can rely on default physical criteria, which would miss, in our experience, some of the smaller streams, which are at times used by salmonids and other native fish that are less than 2 feet bankful width. Same with gradient, 20% is the cutoff. There's been a lot of interest recently in the gradient threshold, above which we won't find fish. Anytime you're talking about gradient, you have to talk about the distance over which it's measured. A 20% gradient over 10 feet is a very different thing than 20% gradient over 200 feet. And so in some of our surveys, we have documented cutthroat trout, for example, in stream reaches in excess of 25% gradient over several hundred feet. It has a lot to do with the habitats present in those steeper stream reaches. If it's a bunch of step pools, for example, there can be fish living within those pools or using that habitat at certain times of the year. So the default physical criteria capture a lot of what we understand to be fish habitat, but there's always going to be exceptions that are exceeding or above and beyond those criteria where fish can be found at certain times of the year.
Great, thank you. I think we have time for one more question, and this might be a doozy. CMER and DNR is updating water types per our HCP requirements. How will conflicts with your water types be resolved?
So CMER and DNR are working together to replace the current water typing rule, which is actually an interim rule. That's 222-16031 WAC, with a permanent water typing rule. This is a process that's been going on for decades, actually, and is likely to take some more time yet. And my understanding is that the water typing done until that new rule is in place will stand. It's essentially grandfathered in, but that remains to be seen. We, Wild Fish Conservancy, and me in particular, are very involved in that process, and we are contributing technical advice to the teams working to finalize the new water typing rule, and we'll continue to be watching that process very closely. We do see the rule moving in the good direction. As I mentioned, right now there's a lot of reliance on the presence of fish on one day to help identify fish habitat, which could be used by fish on any day. And that raises some concerns, and we're working towards a new permanent rule which addresses those concerns.
Great. Thank you, Jamie. And with that, we are at time. So I want to thank you once again for this presentation. It's been really interesting, super informative. And I want to thank everyone who made time for this presentation today. It's great to see all the folks who are able to show up. So I hope you all have a great rest of your day.